So welcome back friends. In this video we will going to talk about inverse PCR, which is a variation of the normal polymerase chain reaction. So before going to talk about inverse PCR, it is important to talk about the normal PCR one more time. So what we are doing in normal PCR? So normal PCR as described by Karimulis in 1983, we are performing selective amplification. So what is the meaning of selective amplification? So the meaning of selective amplification is, suppose this is my gene of interest and it is present in a sample source. Okay, so now with the help of two primers, forward primers and reverse primer, we are performing the amplification of specifically that red marked portion, that is our gene of interest. And after the third cycle we are getting the purified amplified product. That means we are getting the product which are like this, right? So here the important thing was, we know the sequence, that is sequence to be amplified, right? That means we know what is the sequence of our gene of interest. That means the red mark portion is known to us in the basic principle, basic type of PCR. Okay, and with the basis of the 3' in of the known sequence, we are making the forward primer and reverse primer. But in case of inverse PCR, the sequence that we are willing to amplify are unknown. Okay, and it is not in the middle. Instead, it is at the two ends. Okay, and in between them there is a known sequence. Okay, so that is the problem that we will going to solve with the help of inverse PCR. So in case of inverse PCR, the gene of the sequence which is known to us is in between, and the sequence to be amplified is unknown, and they are at the two end. Okay, so how can we do this? So for this thing it is important, because remember, in case of normal PCR, if you try to recapitulate it once again, we are taking the help of the 3' end sequence to create the primer. Okay, so the approach should be to somehow bring this green marked portion at the two end. Okay, so how can we bring them at the two end? Whenever we are talking about bringing a large fragment into two fragment at the two end, what comes in your mind? Your mind should think about the restriction digestion, right? So here we will going to take the help of series of restriction digestion and ligation step. So how we can do this? So we will going to talk about the function of the inverse PCR after learning about the procedure first. Okay, so that is the thing. So in between the two unknown sequence, the known sequence is present, and we will going to bring this known sequence at the two end of the DNA somehow. Okay, and first, for doing this, it is clear that we have to break them, otherwise we cannot bring one molecule at two different sites at the same time, right? So first thing is, we will perform restriction digestion. Okay, and this restriction digestion will be performed with the help of that restriction enzyme that will create a staggered in for us. Okay, so now we will perform the restriction digestion, and remember it, we will try to perform restriction digestion in such a way that our unknown sequence should not be hampered. That means, suppose up to that part we want to sequence, then the genes which are the DNA fragment which are outside the sequence of our interest should be chosen to perform the restriction digestion. Okay, so after the digestion there will be a staggered cut for you. Okay, so now you will perform ligation. Now there is a thing, because in case of experiment we will not use only one DNA fragment, right? There is multiple of DNA fragment. So why they will self ligate instead of ligate with one another? That means I am willing to say that there are too many fragments present. Okay, so if you just add ligase and expect a self ligation, so instead of self ligation there can be also a probability that this fragment will join with this, then this one, then this one, and then this one, and give you a very large DNA fragment instead of self ligation. So we should choose some condition that will promote self ligation, right? So self ligation can be promoted by using low DNA concentration. That is the very important thing, or the condition which is known as quick ligation condition. Okay, so remember, during the ligation step we will use a very low DNA concentration. Okay, so we have circularized it, right? So now, as I have told you that this sequence is very known to us, so it is obvious that we should know that there can be a restriction site present. Okay, so we will use that particular restriction enzyme to digest our known sequence. Okay, and now remember it, we will try to choose such restriction enzyme that will create blunted hair. Okay, so we will get then this thing, right? Because during the restriction mapping videos I have also told you, if there is a one restriction cut site present and you are performing the restriction, then the circular DNA will be linearized, and that is what we have observed there. Okay, and now it will be the normal traditional PCR. You will construct a primer on the basis of the DNA sequence at the 3' end here. Let's say this one is 5' end, and then you will also prepare a primer on the basis of the DNA sequence here, and then you will perform the normal PCR reaction that we are performing. Okay, so that is the concept of the inverse PCR. So I hope you can understand why the name inverse PCR, because here the initially the known sequence was in between. So for performing the PCR well, we are inversing the position of the known sequence. Okay, so again try to recapitulate that. Suppose the red mark portion is your sequence to be amplified, but it is unknown to you. In between them there is a green sequence present. This green sequence is known to you. Then again the unknown sequence, and then the sequence which is not required. So now what we will do? We will do restriction digestion here, right? And during this restriction digestion we should prefer a staggered end generator. Okay, that will provide us staggered end. Okay, now we will perform self ligation. Okay, for performing self ligation along with ligase, it is important to make sure that we have used low DNA concentration. Low DNA concentration, or the condition which is known as quick ligation condition, will promote self ligation. Okay, and due to this self ligation the DNA will be circularized. okay, and then we will perform restriction digestion. Okay, this restriction digestion will linearize it again, and here during that time we will prefer a blunt end cutter. Okay, now we have our known sequence at the 2 end, that is the most preferred format of normal PCR. Then we will generate or construct the primer, and then we will perform the PCR in the thermal cycler machine. So that is the concept of inverse PCR. So in what type of approach we will use inverse PCR? Suppose I think you know about the jumping genes known as transposon or transposable element. So with the help of inverse PCR we can find out where a transposable element can insert in a particular genome. Okay, with the help of inverse PCR. Not only that, we can amplify and identify unknown sequence which are flanked in between, which are flanked at the 2 end of a known sequence with the help of inverse PCR. So we are using inverse PCR for amplifying to unknown sequence at the 2 end of a known DNA sequence. For performing this, we are taking the help of series of restriction digestion and ligation step, and during the first restriction digestion we should prefer a staggered in, then via self ligation. And how can we promote self ligation? We promote self ligation by using low DNA concentration. Then after self ligation we will perform again restriction digestion, that will digest the known sequence, and here the preferred cutter will be the blunt end cutter, and then we will get our preferred orientation of the DNA, with the help of which we can perform the normal PCR technique and get our desired result. So that is the concept of inverse PCR.